So with this chapter, we're looking at blood. So this is exercise 29, and we'll be starting on page 417 in figure 29.1. So this one just shows you the basic components of blood, the plasma versus the formed elements or the cells. Now remember, blood is a connective tissue, so we're going to have the plasma, which is that non-living matrix, and the formed elements, which are the cells. The image here is actually an interesting image. That's what it looks like whenever you do a hematocrit. You may see this in labs if you've ever given blood. Table 29.1 is going to be real important for this one. I could ask you how many red blood cells do we have on an average person? I can ask you what are granulocytes, what are agranulocytes, which white blood cell is going to appear with a parasitic worm, what kind of red or white blood cell is going to not be able to kill, so who is a phagocyte, who is not a phagocyte. So there's a lot of word questions that will come from table 29.1. Plus, as we look at the cells, I might ask you those questions as we point to that cell. Okay. Now, there's a handout online for blood. The handout online is dealing with blood testing. So how to do a white blood cell count, red blood cell count, so on and so forth. So again, know that chart. I could ask you, what tool do I use to do a hematocrit? Or what is the average amount of red blood cells a male is going to have? You will have at least one question where I give you the blood work of a patient and ask you to tell me if they are male or female. And then I'll also need to know if they're sick or healthy or what could be going on with them. So you'll need to know that handout forwards and backwards, along with the information for blood typing within that handout. 29.2 is how to make a slide. Don't do that. You may or may not have microscopes at home, depending on it. I guess if you're an uber nerd, you might have a microscope, but don't do it. So we're just going to be looking at slides of blood cells. So figure 29.3 and 29.4 kind of shows you that. So here's just a typical slide of blood. All the red guys are red blood cells. The little bitty dots are platelets, and the bluish purple guys are white blood cells. Now keep in mind, red blood cells are 7 to 8 microns in diameter, and since they're all over the blood slides, they're kind of like built-in little rulers. So you can compare the size of the white blood cell to the red blood cell to see which one it is. So this little guy is almost identical to the red blood cell next to it, and the only white blood cell that is that small is a lymphocyte. So this is a lymphocyte. With lymphocytes, again, they can be 5 to 17 microns in diameter. The big ones are usually in the lymphatic system. So the ones that we see in blood and the ones I will show you are all going to be small. So that's going to be your first hint that this is a lymphocyte. Another thing with lymphocytes, again, their nucleus is round, and they have very thin cytoplasm. So you'll notice here we barely see any cytoplasm. So it's often described as a halo of cytoplasm. So the nucleus is round and they just have a thin halo of cytoplasm. Because they're agranulocytes, they do not have any granules in their cytoplasm. And these guys do not eat. They are not phagocytotic. This is a monocyte. It is huge. All of your monocytes are big. They start off being the same size as the other white blood cells and just get bigger. They have a huge thing of cytoplasm that is also not going to have granules, so they are agranulocytes, and these are big eating cells. Now these cells, we're going to ignore them for lab, but in lecture we'll talk about bands or rods. These are immature neutrophils. So it's kind of like if you're 17 or 18, you might have a job and technically you're an adult, you can vote, but we don't expect 18-year-olds to have a career and own their own home and have retirement plans and so forth. They're adults, but they're not like a 40-year-old adult. That's kind of how these little immature neutrophils are. They're neutrophils, but they're not really ready to deal with life yet. Okay. This slide, again, we're going to have red blood cells. We have platelets, the little dots, and that has a really big monocyte, so big clear cytoplasm. Here again, red blood cells. We have our platelets, the little dots. And then this guy, this one is almost the same size as this guy. He has a very big nucleus, a little halo of cytoplasm. That is a lymphocyte. This guy and this guy, these are neutrophils. So neutrophils have really loby nuclei, so 3 to 5 lobes. So this one looks like honestly, it looks like a set of kidneys and a bladder. This one looks like he's happy to be here. Lots of lobes, so those are neutrophils. Now you're going to notice we see lots of neutrophils and lymphocytes, and that's because neutrophils make up 45 to 75% of your total white blood cell count. Lymphocytes make up 20 to 45%. So we're going to see these a lot in the slides. And this is just another lymphocyte. Again, red blood cells, platelets. This is huge, okay? A big cytoplasm. And we're going to have that nucleus that's a U-shaped or kidney-shaped. This one looks almost identical to the drawing in your book. That is a big old monocyte, a big eating cell. And there's another neutrophil. Again, neutrophils are going to go around phagocyzing pathogens like bacteria. They're going to be eating debris, so they're a good cleanup crew and first line of defense for the body. Red blood cells, platelets. There's a lymphocyte with his little halo of cytoplasm. That's a mess. Don't look at it. Okay. Red blood cells, platelets. Okay, so here we have our white blood cells. This is a lymphocyte. So again, large round nucleus, little halo of cytoplasm. These two guys are neutrophils. Okay, this one looks like, you know, he just needs a pair of sunglasses to go. So neutrophil. You'll notice this guy is red. This is the only white blood cell that goes red. This is an eosinophil. They almost look like the 7-Up character Spot. Back in the day, they used to have the 7-Up red circle with sunglasses on it. So that is an eosinophil. He's going to be fighting off parasites. So if you have tapeworms or pinworms or fungus infections, they're going to go crazy. They also help with allergy and asthma. So if you've been watching your TV commercials, because it's like you've been stuck at home for a while, they've got new medications for people who have eosinophilic-based asthma and eosinophilic-based allergies. They're suppressing these guys. This guy is a basophil. So you'll notice he's almost the same size as the eosinophil. Same thing with the neutrophil, almost the same size as those guys. Basophils have so many granules, it's really hard to see the nucleus. So they're just chock full of little granules. Their granules are going to have heparin and histamines. So basophils don't eat. This guy is a phagocyte. This guy is a phagocyte, but this guy isn't. He just releases histamines. This is what makes your nose itchy. This is what makes you have runny noses. This is what makes your eyes itchy and irritated. So the Benadryls usually are going to suppress histamines, and that's these guys here. So the histamine and heparins, they're there to irritate the tissue, so to speak, to draw more blood. And more blood means more white blood cells to fight off an infection, more eosinophils to fight off an allergen. So we'll see these guys if we have a lot of inflammation, whether it's an allergy inflammation or an infection. You know, you've scratched your arm and now you've got an infection inflammation. So red blood cells, platelets. There's a monocyte. It even looks like an inframonocyte, but huge cytoplasm with no granules. There's an eosinophil, again, the only guy that goes red. We have red blood cells. We've got platelets. We've got this screaming looking neutrophil over here. Okay. Figure 29.2, again, we're not doing our own blood. Don't look at it. 29.3 and 4, we've just gone through. When we look at the different testing, the first thing that we can use for blood testing is a hemocytometer. So it's a special slide used to count blood cells. You can actually kind of see it with the photograph. It has a grid built in on it, and there is a math problem you don't have to know, that you count how many red blood cells are in this grid, and you predict how much you have in the blood and how many white blood cells you have in the grid. So the hemocytometer is used for red blood cell counts and white blood cell counts. When you have the handout of the blood tests, this is the hemocytometer. This is what you use for those first two blood tests. Okay. Figure 29.5 is just showing you the way to do a blood count without using a hemocytometer. It's a little bit more difficult. 29.6 shows you the steps of determining a hematocrit or packed cell count. So you put your blood in a tube that is not going to allow for coagulation, and then you put it in a centrifuge and it separates out the plasma from the red blood cells. So you'll have plasma on top and red blood cells will sink to the bottom. It looks exactly like the test tube on figure 29.1, and that's your packed cell count. The next image is using a hemoglobinometer, so 29.7. That is the old school version of a hemoglobinometer that's completely manual. So what you would do is you would take a drop of blood, you would put it on the special applicator, and you would break it down. You would do hemolysis that would release the hemoglobin, and then you would put it into the hemoglobinometer to count the molecules of hemoglobin. This is an electronic hemoglobinometer, so you just put the blood sample in and the computer counts it for you, so you don't have to do the physical counting. So that hemoglobinometer is used to determine how much hemoglobin an individual has. Again, this is a tool that you would use for the blood testing. Coagulation time, we do talk about that in the chart. So you just put your blood in a non-herpetized capillary tube that looks just like the tubes that you see in figure 29.6. And then at 2 minutes, you break it in half and pull. If it has a coagulation, it'll look like mozzarella cheese sticks after you break them. You've coagulated, you just record the time. If you haven't coagulated, there won't be a string between the two halves. And then you just continue to break the test tubes. You take several. And then determine when the person coagulated. Figure 29.8 shows you what coagulation looks like step by step. This is something that will be more important for lecture than for lab, so you can kind of ignore that. Blood typing, figure 29.2 or table 29.2, excuse me, shows you one version. There is a handout. Again, when you print the handouts for this lab, that is going to be the numbers I will ask of you. So you need to know the percentage for each blood type, A, B, and O, as well as for positive, negative, so RH factors. 29.9 shows you some samples of doing blood testing and what it will look like. So that's what 29.9 is showing you. Instead of doing this in the lab, what I would like for you to do is to go to the Nobel Prize site. Okay, so you can find that if you go to your Blackboard. All right, so I want you to go to your content, go down to Learning Games, and then the blood typing game, and this will take you to the Nobel Peace Prize site. You can actually go to this site directly if you want. It's nobelprize.org, and there is a blood typing game. I want you to play at least one round. So do a quick game. This will give you 3 patients. After you're done playing, I want you to take a screenshot of your results and put that into the blood game Dropbox, and that will count for your lab list for the blood chapter. And the neat thing about this game, as you're going through, it's giving you hints on how to identify blood typing, and it gives you a lot of good information and tutorials at the end of the game. So content, learning games, blood typing, play the quick game and screenshot the final results and drop that into the Dropbox for this week's Dropbox assignment. All right. And that is it for the blood lab.